Well, aloha everyone, and welcome back to Anatomy 2 here at Chaminade University. Today we will be discussing the endocrine system. So the nervous and the endocrine systems are actually a coordinated mechanism of maintaining homeostasis for all of the systems of the body. So while the nervous system, as we just spoke about, releases neurotransmitters, so things like acetylcholine, etc., the endocrine system is going to get to their target organs by way of hormones. Another major difference is that the nervous system and neurotransmitters, etc., are going to be in a very localized capacity. So when we have the end of an axon interacting or synapsing with another neuron, it's a very localized release of the neurotransmitters. Whereas hormones are more of a global effect. They're going to be released into circulation and circulate throughout the entire body. As I mentioned, they're going to circulate through the bloodstream, and then they bind to target cells by binding to specific proteins on the outside of the cells that are known as receptors. So neurotransmitters also work this way. They bind to receptors on those target cells, but as I mentioned, that's going to be more of a localized instead of being in broad proximity. So they're going to be much closer to their actual targets when released. So this here is an overview comparing the nervous system and the endocrine system. The mediator molecules in the nervous system are neurotransmitters. In the endocrine system, they're hormones. And again, neurotransmitters are going to be released locally, whereas the hormones are going to be released more globally. So the site of mediator action in the nervous system is going to be much closer to the site of release, namely at a synapse, where it's going to bind to the receptor molecules at that membrane and that postsynaptic region in the junction between two neurons. Whereas in the endocrine system, generally, the mediator action site is far away from the site of the release. So while we're binding to receptors on or in target cells, we're doing that from a much further distance away. Now, the types of target cells vary. In the nervous system, it can be anything from muscle cells, like your smooth muscle, cardiac, skeletal muscle cells, gland cells, other neurons, etc. In the endocrine system, it's going to affect many different types of organs and cells throughout the body. Generally, the nervous system is going to react much more quickly, so within milliseconds. If you think of a nerve firing, the action potential is almost instantaneous, and its passage is almost instantaneous, so it's going to only last for milliseconds, whereas the endocrine system is going to take a little bit longer to have the onset of action, so it takes a little bit while to release the hormone, to have the hormone circulate, to find its target cell, etc. And because it's going to be released globally, it also is going to take a little while for it to dissipate, so generally the duration of action is going to be longer as well. Now, exocrine glands are going to be anything that's going to secrete their product through a duct that is non-hormonal. Again, a non-hormonal secretion is through an exocrine gland. Things like sweat glands, oil glands, mucus glands, and digestive glands are all examples of exocrine glands. And again, they have ducts. They're going to secrete something through that duct into another medium. If you consider your digestive glands, for example, they would be secreting intestinal juices through those intestinal glands into the lumen of the intestines. Now, endocrine glands are a little bit different in that they secrete hormones. They are also ductless, so they do not have any ducts, and they secrete their hormones directly into the interstitial fluid. So to just backtrack a little bit, you've got your blood, right? Your blood is then going to release things into the interstitial fluid, and that interstitial fluid is going to then exchange things with the cytoplasm of the cell. So while all three of these are liquid media, they're going to vary just slightly in what they're comprised of, right? Concentrations of gases and salts and etc. So basically, things are going to come from the bloodstream into the interstitial fluid, from the interstitial fluid into the cytoplasm in terms of, say, delivering groceries to the cell or biomolecules the cell needs. And then they're going to go out in terms of cellular waste and things the cells want to off-gas from the cytoplasm into the interstitial fluid into the blood supply. And so hormones are going to take advantage of that, diffusing into the bloodstream, carried through the capillaries, and then end up at different target cells throughout the body. And we have many different endocrine glands. The endocrine glands that we're going to discuss today include the pituitary gland, the thyroid and parathyroid glands, the adrenal gland, which we touched on a little bit when we talked about the autonomic nervous system, and the pineal glands. We also have organs and tissues throughout the body that secrete hormones but are not part of the endocrine system. Those are to include things like the hypothalamus, the thymus, the pancreas, the ovaries and testes, kidney, stomach, liver, small intestines, skin, heart, fat tissue, and placenta. Now, placenta is interesting because it is an organ, but it is a temporary organ, so it's it is the only temporary organ. All right, so this is an overview of where the endocrine glands are located in your body. Here we have the thyroid gland, which is going to be found at the very top of the throat, above the trachea. The parathyroid glands are found just posterior to the thyroid gland, so behind the thyroid glands. We also have the adrenal glands, which are located on top of the kidneys. Now remember, the adrenal glands have both a cortex and a medulla, and they do different things. We talked about the cortex previously, and we talked about the medulla previously, and we talked about norepinephrine release, and we'll be talking about the cortex today. In the brain, we have the pineal gland, the hypothalamus, and the pituitary gland. And the hypothalamus and the pituitary gland are connected by something called the infundibulum. We'll talk about that when we get there. We also have the ovary and the testes, which are going to be secreting hormones as well. Now, as I mentioned, hormones are going to have kind of a global effect. They're going to travel throughout the body, through the bloodstream, until they reach whatever their target cells are. And the way that they're going to interact with those target cells is by binding to a protein receptor. So there's a specialized receptor on the surface of the cell. Generally, there's going to be several of them, hundreds if not thousands of them, that are specialized for whatever that hormone is. And when that is bound, then it's going to trigger a pathway. Now, receptors are constantly being created and destroyed. And in this way, we are able to vary both the concentration of the hormone in circulation and the concentration of receptors on the cell surface. So we're able to very tightly mitigate the interaction of these two. We're able to both up or down regulate a receptor, depending on the concentration of the hormones. We can basically fine tune our signal. And that's going to allow us to be able to very tightly hone in on the pathways or the intracellular cascades that occur when these hormones bind. Okay, so this is a very childish cartoon, basically, of how hormones are circulated in the body. So we have an endocrine cell here. This endocrine cell is going to secrete a hormone, which is going to end up in the bloodstream. So basically it comes out of the cytoplasm, released into the interstitial fluid, from the interstitial fluid into the bloodstream, where it enters into circulation. Then it travels through the capillaries. Capillaries are notoriously very leaky. Things are going to extravate out of them. And then they're going to find their target cells where they're going to bind to a receptor. And this is a very simplified diagram, little ball with a little socket. But really what we're going to be seeing here is more along the lines of a lock and a key, very specialized three-dimensional structures that are only going to interact one hormone and one receptor, right? So receptor A, receptor B, hormone B, etc. Now, depending on what type of hormone, this hormone might end up binding to the surface, or it might ending up inside of the cell, affecting some sort of intracellular cascade. Now, some hormones are actually going to interact on a more local level. So instead of acting on that global level that we just saw, and so previously we were talking about an endocrine cell, right? These are going to end up going into the bloodstream and circulating. But we also have other types of hormone secretion. So we have hormones that don't circulate very far. Those are called paracrine. They're more localized. So if you were to picture that that this endocrine cell that we just talked about is the mail delivery service for the United States, right? And it's going to be a global mail delivery service. This local will be more along the lines of your local apartment complex putting flyers in everybody's mailbox, right? So it doesn't go very far, and it's just going to hit local targets. But we also have cells that are going to secrete hormones that are going to act on themselves, and those are called autocrines. So there's an autocrine loop, basically, where a cell can secrete information that then it is going to receive by its own receptor that triggers its own intracellular cascade. And this would be more along the lines of you sending yourself a note. Tomorrow I need to go to the grocery store or whatnot. So it's basically informing only a very, very localized subset, mainly just that one cell. As I mentioned, hormones are biomolecules. And biomolecules come into major classes. One of the ways that we divide biomolecules is into lipid-soluble or water-soluble. So basically hydrophobic and hydrophilic. And hormones that are lipid-soluble include steroid hormones, thyroid hormones, and also nitric oxide, which is a gas. And hormones that are water-soluble include things like amine hormones, peptide and protein hormones, and eicosanoid hormones. Now, as you might imagine, because water is aqueous, right, and blood is an aqueous solution, water-soluble hormones are going to circulate freely in the blood plasma, whereby the lipid-soluble proteins are going to need a transport protein to help carry it. So you can picture this like the grease that's on your pan, right? It's not just going to come off in the water. It's not going to interact well with the water. It's hydrophobic. So what you need is something that's, the word is amphipathic, and that means that it has one region that's hydrophobic and one region that's hydrophilic. A good example of this would be a soap molecule. So a soap molecule will pull that grease right off the pan and allow it to interact with the water, because basically it forms globs around the grease where all the regions that are hydrophobic bind to the grease, all the regions that are hydrophilic bind to the water, and it pulls the grease off of the pan. So there's a carrier molecule that allows it to be transported through the bloodstream. So lipid-soluble hormones do circulate through the bloodstream, but they're bound to transport proteins that basically hide the fact that they're hydrophobic. Now, as usual, I'm never going to ask you for the structures of these, but just so you're aware, the steroid hormones do have, have that basic setup there where we have 3 6-carbon rings and 1 5-carbon ring. And an example of that would be aldosterone. We also have cortisol and other androgens, which are going to be the male-specific hormones. Things like calcitriol, which is involved in calcium. That's going to be secreted out by the kidneys. Testosterone, which is secreted by the testes. Estrogens and progesterone by the ovaries. And then thyroid hormones, which are going to come from the thyroid. That's T3 and T4. They come from the follicular cells in the thyroid gland, and we'll get into them in just a minute. And last but not least, another example is a gas. So this is a hormone, but it is a gas. It's nitric oxide, and it's secreted by the endothelial cells that line the blood vessels inside the body. We also have some water-soluble hormones, which include your amines, like epinephrine and norepinephrine. You may remember these when we were talking about the autonomic nervous system. Again, these are secreted from the adrenal medulla, specifically from the chromaffin cells. Things like melatonin, which is involved in sleep and the sleep cycle, that's secreted from the pineal gland. Histamine, which anyone who has had any sort of allergic reaction or allergies understands, right? You take Claritin, that's an antihistamine that helps slow down the autoimmune response when something is entering into your body, the immune response. And that's secreted, histamine is secreted by the mast cells in your body. And serotonin is like a nice feel-good molecule secreted by the platelets in your blood. All of these are good examples of amine-style hormones. Other types of hormones include peptides. And here's just an example of the amino acids in a particular structure. A good example of a peptide or a protein hormone would be oxytocin, which we know of as the love hormone. And so all of the hypothalamic releasing and inhibiting hormones, which come from the hypothalamus, are going to be peptide and protein hormones. Now, we'll talk about these specifically in a minute, but releasing and inhibiting hormones are simply hormones that are going to act on the concentration of other hormones. So if it's a stimulating hormone, it's going to increase the amount of that other hormone that it's acting on. If it's an inhibitory hormone, it's going to decrease the amount of the hormone, whatever hormone it is it's acting on. And we'll talk about specifics of that in just a minute, but just to kind of let you know that these aren't really going to be doing anything in and of themselves except regulating the concentration of another hormone that has a function in the body. Another example, or other examples include oxytocin, that love hormone when you are bonding as a mother with a breastfeeding child or something like that. Antidiuretic hormone, which is involved in making sure that the body holds onto the water. So coffee being a diuretic, if you were to consume 20 ounces of coffee, you tend to urinate out more than 20 ounces of coffee because as a diuretic, it helps remove water from the system. Antidiuretic hormone would help retain water, so it would tell the kidneys to concentrate the urine and try to bring the water back into the body. That's released in the posterior pituitary. In the anterior pituitary, we release several different hormones, including HGH, or human growth hormone, thyroid stimulating hormone, which does exactly what you think. It stimulates the activity of the thyroid. Adrenocorticotropic hormone, follicle stimulating hormone, luteinizing hormone, prolactin. Those last 3 are involved in the reproductive pathways. Melanocyte stimulating hormone, which is involved in the darkening of the skin in response to UV light to help create tan skin. Insulin, glucagon, somatostatin, pancreatic peptides, all of these are secreted by the pancreas and are involved in digestion or in regulation of blood glucose. Parathyroid hormone secreted by the parathyroid glands. Calcitonin, which we'll talk about in a little bit, almost pretty extensively, and that's going to be involved in regulation of the blood calcium levels. That's secreted by the thyroid gland, or particularly the parafollicular cells of the thyroid gland. Secreted by the stomach, we have gastrin, secretin, cholecytokinin, GIP, which is glucose-dependent insulin-trophic peptide. We're not going to quiz you on that. But all of these are involved in the feeling of hunger and the satiety or the feeling of satiation or the initiation of different blood glucose levels, etc., during digestion. So they're all going to be involved in digestion and the feeling of needing more nutrients or of being full and absorbing the nutrients, etc. They're secreted, again, in the stomach and small intestines by the enteroendocrine cells. The kidneys are going to secrete something called erythropoietin, which you may recognize the word erythro from erythrocyte or red blood cells. So this is going to help create or stimulate the creation of blood cells. And in the adipose tissue, well, it's going to secrete the hormone leptin, which is also involved in the feeling of satiety versus hunger. And we also can secrete things like prostaglandins and leukotrienes. Again, those are going to be another type of water-soluble hormone, but in this case, we've just entered into the echinosoids. So we just finished the peptide and protein list and entered into the echinosoid list. All right, so different cells are going to vary in their response to the hormone, and it's going to do this because of a lot of different ways, right? So some of the responses that a hormone may initiate would be the synthesis of a new biomolecule, right? Initiation of a cascade that creates a new molecule. It can also initiate a cascade that changes the permeability of a cell membrane overall or to a specific molecule, right? A specific molecule wants to bring more in or send more out, which leads me to the next one, stimulating the transport of a particular substance in or a particular substance out. And that goes hand in hand with the ability to alter the rate of the metabolic reactions within the cell. And another response that's fairly typical if you're a smooth muscle cell or a cardiac muscle cell would be the initiation of a contraction of that cell. Now, depending on whether you are lipid-soluble or a water-soluble hormone, you're going to have a different mechanism of hormone action. Now, the first thing I want to draw your attention to here, we're dealing with the lipid-soluble hormones, is that these guys are going to be transported by specific transport proteins, so it looks like a blue wrench up on the screen. You can picture this the equivalent of, remember my previous example of grease and soap, right? So we have a hydrophobic molecule, or this little green free hormone, and that would be the equivalent of like grease, right? And it's going to be transported by that amphipathic molecule, this transport protein, that allows it to go through the blood supply, so it interacts well with aqueous solution, even though it itself does not. And then it's going to somehow extravate out of the blood vessel and diffuse into the cell. Now, because it's lipid soluble, it has no problem going through that plasma membrane. It can get right on in the cell, not a problem. Not only can it get right through that plasma membrane, guess what? This nuclear membrane, also going to be lipids, so no problem, slides right on through. That means that it can get all the way to the DNA. And in this way, it's able to up or down regulate gene expression. So it's going to actually go directly to the source and change it from the DNA level up. And it can do that, again, by making more or less DNA, which leads to more or less RNA, which then leads to more or less protein. Now, water-soluble hormones are a little bit different. Water-soluble hormones are not going to go right on through the membrane of the cell. So while they are going to extravate easily out of the blood capillary, they're going to have to bind to a receptor. So there'll be a receptor on the outside of the cell. We talked about how receptors and ligands work. In this case, the ligand is this water-soluble hormone. And it's going to initiate an intracellular cascade. So instead of going all the way into the house and talking to the boss, it's going to knock on the door and talk to the butler. And then the butler is going to talk to the secretary, and then the secretary is going to pass that signal on. In this case, the butler being the receptor and the first secretary being a G-coupled protein receptor. So that G protein is going to interact with adenylate cyclase, which causes the conversion of ATP into cyclic AMP. Cyclic AMP is a second messenger molecule. And again, you're not going to need to know every step of this cascade, but just be aware that multiple steps are going to happen, eventually leading to the activation of protein kinases and the phosphorylation of particular proteins. Now, depending on which protein is phosphorylated and how many of them, they are going to initiate different physiological reactions. So typically, phosphorylating a protein activates it. Sometimes it deactivates it, but typically it activates it, and so that's going to mean that it's going to be able to perform whatever its physiological function is. So in this way, we're able to initiate an intracellular cascade by knocking at the front door and not actually entering the cell. Okay, so the target cells themselves have the ability to respond to a hormone in different ways as well. And that's going to be based on several factors, including the concentration of the hormone in the blood, right, just in general circulation, which we can up or down regulate based on how much we need, so we can release more or release less. And then also based on the number of hormone receptors on the target cell. So remember I told you we're constantly up and down regulating the amount of receptors. So sometimes we might have more, sometimes we might have less. Certain cells might have, say, 1,000. So say you're a cell that only has one, right? If it's bound, you're 100% bound. But if we have 1,000, you could have 20 out of 1,000, which would be minimally bound. You could have 50 out of 1,000, a little bit more. You could have 500 out of 1,000 bound. And in that way, we would have different ways of regulating the cascade based on the concentration of the hormone receptors and the hormone concentration in the blood. Additionally, other hormones can also exert an influence on a particular hormone. So for example, remember I said this guy comes up and knocks on the door and tells the butler a message. So say a hormone comes up and knocks on the door and says, there's a fire down the street, right? Now, you as the individual have to make a decision whether or not to evacuate. Now, a second person comes and knocks on the door and says, it's okay, it's just my barbecue. You might go, never mind, I'm not going to evacuate, right? That would be called an antagonistic effect. One's going to work in opposite of the other. One person saying you should evacuate, and the other guy's going, nah, don't worry about it. So you'd be more reluctant to pack everything up. On the other hand, if the first guy came and knocked on the door and said there's a fire down the street, and the second guy came and knocked on the door and frantically said, there's gasoline all over the road, you need to evacuate right now, there's a gas leak, right? That, on the other hand, would be considered a synergistic effect. That second knock would actually be more likely to make you leave the house than the first one. So sometimes the sum of the two is actually more than it would be of either of them individually. And sometimes they work against each other. So again, that's synergistic if they're working together, and antagonistic if they are opposing the actions of other hormones. Generally, hormones are secreted in short bursts and only when needed, and it's regulated on many different levels based on signals from the nervous system, other hormones in the regions, and chemical changes in your bloodstream. Now, you've probably seen these negative feedback loops before. I've certainly gone over them before in 151, but if this is your first one, welcome. Typically what happens here is we have a stimulus that gets disrupted. We have a controlled condition that gets disrupted by a stimulus. In this case, the controlled condition is blood pressure, and it has been increased by a stimulus. Now, the detection of that controlled condition, in this case, there's always receptors that detect the condition. In this case, those receptors are baroreceptors in the blood vessels. And then they send information to the control center, which is the brain, which has an effect. It has an effect on the heart and the blood vessels, and that effect leads to a response, which is a decrease in the heart rate, and also a dilation of the blood vessels, which is going to cause a decrease in the blood pressure. And once we've returned to our homeostatic norms, then we're going to shut this off, right? So we've then returned back to homeostatic conditions. That's considered a negative feedback loop. Now we also have positive feedback loops, and these are much, much fewer and farther between. Usually a positive feedback loop happens when we need to have a particular course of action taken. Like we have to deliver a baby, or we have to remove a splinter from our body, right? We'll push that splinter forward, right? So a positive feedback loop is when disrupting the condition causes further disruption of the condition. So an example, again, so the delivery of a baby. So the stretching of the cervix, right? The controlled condition is the stretch of the cervix. It starts to get more stretched, which causes the brain to send information to the wall of the uterus to contract. Contraction causes more stretching. Stretch is causing more contraction. Contraction causes more stretching. And the only way to end this positive feedback loop is by interruption of the cycle, delivery of the baby, for example. So again, these are very few and far between, and they usually are in very unusual situations where you really are trying to return to normal, right? You're trying to return to pre-baby conditions, in which case you have to have something extreme happen in order to push through to be able to get to the other end. Again, most of our cycles are going to be negative feedback cycles. Certainly everything that we look at today is going to be negative feedback. All right, so let's switch tracks a little bit, talking about the hypothalamus and the pituitary gland. So the hypothalamus and the pituitary gland are going to work hand in hand to control a lot of the other endocrine glands. And they're connected by a region called the infundibulum, which is a really fun Scrabble word. And that looks kind of like this. So here we have the infundibulum, which connects into the anterior pituitary. Here's also the posterior pituitary. And the anterior lobe, so the anterior pituitary, makes up most of the weight of the pituitary gland, and it secretes 7 different hormones. The posterior lobe is made of neural tissue and releases hormones that are made by the hypothalamus. So again, the posterior lobe does not create any of its own. It only releases hormones that are secreted from the hypothalamus, stored in the posterior lobe, and then it's going to release it into circulation. So the hypothalamus releases or secretes hormones that are both releasing and inhibiting. So anytime you see something ending in RH, it stands for releasing hormone. If you see something ending in IH, it stands for inhibiting hormone. They're usually going to be antagonistic, right, that they're working against each other. So the releasing hormone is going to release it and the inhibiting hormone is going to decrease it. And these two types of hormones are going to control the release of hormones by the pituitary gland. So the hypothalamus directly is in control of the pituitary gland by way of these releasing and inhibitory hormones. And they're going to get to the pituitary gland from the hypothalamus from the hypophyseal portal system. All right, so this is what I just showed you when we're talking about the infundibulum, the anterior pituitary, and the posterior pituitary. And if you look at where it's actually found in the brain, it's actually going to be found just above the midbrain. So if we did a little bit of a cross-section here and zoomed in, now you'll see here we have the hypothalamus. This is the infundibulum. Here's the anterior and the posterior pituitary. They're going to have their own blood supply that's going to also allow the hypothalamus to talk directly to either of these guys as well in terms of secreting those releasing and inhibiting hormones, which then are going to in turn tell the posterior or anterior pituitary to either release or inhibit release of the particular hormones. Again, that's going to happen through the hypophyseal portal system. So these are the hypophyseal portal veins, the hypophyseal portal artery. All those are going to wrap right around the infundibulum. All right, so the anterior pituitary gland secretes 7 different hormones. And these hormones are produced by 5 different types of cells. The first that we're going to talk about is human growth hormone. It's also known as somatotropin, which is why the cells that are secreted are labeled somatotrophs. And the next column is going to tell you what's going to stimulate the secretion and what's going to suppress the secretions. That's what the next columns tell you. So we can see that human growth hormone is going to be stimulated by growth hormone releasing hormone, and it's going to be inhibited by growth hormone inhibiting hormone. So once you start understanding what IH and RH stands for, it starts to become pretty simple. A releasing hormone is going to increase the amount of that hormone in circulation, and an inhibiting hormone is going to decrease the amount of whatever that hormone is in circulation. Another one released by the anterior pituitary is TSH, or thyroid stimulating hormone. That comes from thyrotrophs, and it is going to be stimulated by thyrotropin releasing hormone and inhibited by growth hormone inhibiting hormone. Follicle stimulating hormone and luteinizing hormone are both involved in reproduction. They're secreted by gonadotrophs, and they're going to be stimulated by gonadotroph releasing hormone. There is no inhibitors. Prolactin are secreted by lactotrophs. This is a hormone that's going to be involved in the secretion of milk by the mammary gland, so in breastfeeding, and it's going to be secreted by prolactin releasing hormone and inhibited by dopamine, or prolactin inhibiting hormone. ACTH is known as corticotropin, or adrenocorticotropic hormone. It's secreted by corticotrophs, and it's going to be stimulated by corticotropin releasing hormone. Same with melanocyte stimulating hormone, or MSH, secreted by corticotrophs and released by corticotropin releasing hormone. Again, the inhibition here is going to be by dopamine. Okay, so I told you that we had feedback loops. What's interesting about several of these feedback loops is that the end result of these feedback pathways are actually the inhibitors of the beginning of the pathway, and this is a good example of that. So here's a negative feedback loop that controls the secretion here of corticotrophin. So here's corticotrophin releasing hormone, or CRH, which is going to be released by the hypothalamus. Now that releasing hormone being released by the hypothalamus through the infundibulum is going to end up increasing the amount of corticotrophin that's released by the anterior pituitary. Corticotrophin goes directly to the adrenal cortex. Remember, previously we talked about the adrenal medulla when we talked about the chromaffin cells. Now we're talking about the adrenal cortex. The adrenal cortex is going to secrete cortisol. And cortisol, as the end of this pathway, is actually going to inhibit, so elevated cortisol levels inhibit the release of the releasing hormone. So in this way, it kind of self-stabilizes, right? Because when we end up with less cortisol, we have less inhibition, which makes more cortisol. More cortisol makes more inhibition, which makes less cortisol. So in this way, we have a feedback loop whereby the end product of the pathway is actually regulating upstream. Now, by far the most plentiful hormone in the anterior pituitary is the human growth hormone. It's released every couple of hours by somatotrophs, and it's controlled again by two hypothalamic hormones, including growth hormone releasing hormone and growth hormone inhibiting hormone. And as you might imagine, because one's a releasing hormone and one's an inhibiting, these guys have antagonistic effects on one another. Okay, so this is an example of two feedback loops kind of intertwined. And we're talking here about the regulation of blood glucose. So if we have low blood glucose, that's called hypoglycemia. If we have high blood glucose, that's called hyperglycemia. If you were just picture that an individual were to eat a bunch of sugar, they would be hyper, right? That hyper is higher, hypo is lower. So anyway, let's start with one. If we have a low blood glucose, that's going to stimulate the release of GHRH, right? And if we have GH, so growth hormone, releasing hormone. So releasing hormone means we're going to secrete growth hormone. So that's going to go to the anterior pituitary and secrete more human growth hormone. When we release more human growth hormone, that's going to enter into the system and also create IGFs, or insulin growth factors. HGF and IGFs are going to speed up certain metabolic processes, including the breakdown of glycogen, which is found in the liver, into glucose, which is going to enter the bloodstream. As soon as it enters the bloodstream, that's going to increase the blood glucose level. It might even increase the blood glucose level to a higher level than necessary, creating hyperglycemic conditions. And when we get into hyperglycemic conditions, that's going to stimulate the release of growth hormone inhibiting hormone. And then growth hormone inhibiting hormone is going to inhibit the amount of human growth hormone that is being secreted by the anterior pituitary, which is going to lower the levels of HGH and IGFs. It's going to slow the rate of glycogen breakdown. Therefore, we're going to have less blood glucose in the blood. When our blood glucose levels fall to the normal levels, we've reached homeostatic conditions. And if they fall too far, we lather, rinse, repeat back to 1 to help stimulate them to go back up again. So in this way, we have two interlocking pathways where we have a releasing hormone and an inhibiting hormone, which both regulate one another such that we can maintain homeostatic conditions. Too high, we bring it back down. Too low, we bring it back up. So to summarize, the anterior pituitary gland is going to secrete several hormones, including HGH, TSH, FSH, LH, prolactin, adrenocorticotropic hormone, and MSH. And yes, you are going to be responsible for knowing what all of those acronyms stand for. Okay, so this table summarizes the hormone, the tissue that that hormone acts upon, and the principal actions of that hormone. The first one in the list is human growth hormone. We also know it as somatotropin. Its target is the liver, and as we just mentioned, it has involved in the glucose concentration. It's going to stimulate those insulin-like growth factors, which promote cell growth, protein synthesis, and tissue repair, as well as the breakdown of fats and the elevation of blood glucose. Now, thyroid-stimulating hormone, which is known as thyrotropin, or TSH, is going to target the thyroid gland. And the thyroid gland sometimes has two lobes. In some individuals, we have this third pyramidal lobe. But that's going to stimulate the secretion of thyroid hormones by the thyroid gland. Then we have FSH and LH. These act on both the ovaries and the testes to help either initiate the development of oocytes or in the testes to produce sperm. In females, LH is going to help secrete estrogen and progesterone, which is going to help regulate the process of ovulation and menses and the formation of the corpus luteum, which is what's left inside the ovary after the egg has ruptured from the follicle. And in males, it's going to help produce testosterone. Prolactin is primarily a female hormone. It's going to help with milk production in the female mammary glands for breastfeeding the babies. Corticotrophin is found in the adrenal cortex, and it stimulates it targets the adrenal cortex, and it stimulates the secretion of glucocorticoids by that adrenal cortex. We also have melanocyte stimulating hormone, or MSH, which is involved in the darkening of the skin in response to UV light. So it's what helps you get tan as opposed to getting sunburned. Okay, so the posterior pituitary gland, as I mentioned previously, does not actually synthesize any hormones itself. Instead, it stores hormones that are created and secreted into the posterior pituitary gland by the hypothalamus. So those are going to be oxytocin and ADH, or antidiuretic hormone. Again, they come from the hypothalamus through the hypothalamo-hypophyseal tract. That looks kind of like this. So again, this is our infundibulum right here, which is going to connect our hypothalamus to the posterior and the anterior pituitary glands. And there's neurosecretory cell bodies that are going to secrete these hormones into the posterior pituitary, where they're going to accumulate until they are induced to be released. And that's going to be the hypothalamo-hypophyseal tract, which is all of these axons right here running from the neurosecretory cells into the posterior pituitary. All right, so ADH, or antidiuretic hormone, is going to be involved in regulating the osmotic pressure of the blood, which is kind of a difficult thing to describe, but basically the concentration of solute in your liquid and water, right, but in the case of blood would be in your blood, is going to be varied from one liquid medium to another. And if you have two different liquids on either sides of a membrane, the amount of what we call osmotic pressure is directly correlated to the concentration of solute in that liquid. So how much free water is available versus how much other stuff is in that water. And that's your osmotic pressure. And the higher the difference between the two, the higher the osmotic pressure in one direction or the other. And so that's how we're going to be able, for example, to make urine, etc. And we'll talk about that in a little bit later, specifically when we get to the kidneys. But the point here is that the amount of ADH secreted is going to be dependent on the blood volume. So basically, ADH decreases your urine output and helps you retain water, which is something that is vital if you're really low on water. But if you have a lot of water, you don't necessarily need this much ADH. And so we have receptors inside our hypothalamus that are constantly monitoring what's called the osmotic pressure of our blood, or really the concentration of free water that's in our blood. And if we have an increase in blood volume, that means we probably have a good bit of free water in our blood, right? It's probably a little bit more dilute. We probably drank more liquids. That's going to cause a decrease in ADH secretion. But if we have a lower blood volume, that's going to cause an increase in ADH secretion. That's going to tell the body, hey, we really need to conserve water and not release water in our urine or our sweat because we don't have enough blood volume at this current moment to meet our needs, or we're running low. Okay, so here's another example of the feedback loops. So if, for example, we have high blood osmotic pressure, remember all of these start with a controlled condition that's been disrupted by some sort of stimulus. In this case, the controlled condition is the osmotic pressure, and a stimulus of some sort has increased your blood osmotic pressure. And the first mechanism that has to happen is we have to detect that change. So we have all these receptors throughout our body that do different things. In this case, osmoreceptors are going to be responsible for detecting the osmotic change. Now, osmoreceptors are going to activate the neurosecretory cells that synthesize and release antidiuretic hormone, and we're going to get firing from nerve impulses to release that ADH from those axon terminals in the posterior pituitary, so from the posterior pituitary into the bloodstream. That's going to have the effect of three major target tissues, including the kidneys, which are going to then be told to retain more water. Again, that's going to decrease your urine output and increase urine retention. And it's going to tell your tissues, like your sweat glands, to stop perspiration, because we don't want to lose that water through sweat. And it's going to constrict your arterioles, which is going to increase blood pressure. Right? It might get to the point where all of this is going to end up getting to the point where our osmoreceptors are now needing to be inhibited. And so we can get to that point if we have all of our target tissues have resumed enough water, et cetera, that now we have our blood osmotic pressure back to normal, right? And that's going to inhibit those hypothalamic osmoreceptors, and inhibition is going to reduce or stop that ADH secretion, right? So in this way, if we go too far one direction, we go back to homeostasis, and again, too far the other direction, we come back to homeostasis as well. Okay, So the posterior pituitary, by contrast to the anterior pituitary, only has two hormones that it secretes. The first is oxytocin. Again, this is a love chemical. This might be something that induces intimacy or feelings of affection post-coitus or during breastfeeding. And it's going to be involved in the contraction of the uterus as well. So it's going to stimulate contractions in the muscles of the uterus during childbirth, and also going to be involved in the process of milk ejection in the mammary glands, usually in response to a stimulus such as your child crying, or perhaps the crying of another child at the grocery store. And it's being controlled by the hypothalamus. So there's neurosecretory cells in the hypothalamus that are going to secrete that oxytocin, and that oxytocin, again, it's going to get stored in the posterior pituitary and eventually end up secreted into the blood supply in response to uterine distension, so like stretching of the uterus, or something like stimulation of the nipples for ejection of milk. The second one, again, is ADH, or antidiuretic hormone. It's also known as vasopressin, and I apologize for this not coming up clearly. I'm not sure what happened here, but again, kidneys, sudoriferous sweat glands, and arterioles. And the objective of ADH basically is to conserve the water in the body. It does that by decreasing your urine volume, decreasing sweating, and increasing your blood pressure by constriction of your arterioles. Because oftentimes if you have a high osmotic pressure, it means you have low blood volume, which means you might have low blood pressure. And again, it's going to be controlled by the neurosecretory cells of the hypothalamus, which secrete the ADH. And they do that in response to particular receptors that say that we have elevated osmotic pressure of the blood, which often indicates that we're bordering on dehydration, and generally comes hand in hand with loss of blood volume. Pain or stress can also induce this. And inhibitors are going to be things like low blood osmotic pressure, so basically the flip side of the coin, high blood volume. But another one is also alcohol. So alcohol is something that can inhibit ADH, which might mean that you might end up with thinner blood. And you may have heard that people who cut themselves while they are under the influence of alcohol can end up bleeding more profusely than others. Oftentimes that's a couple reasons, but it might have to do with the fact that they are not going to be having the right amount of ADH in their system. So they're not conserving that water the way they might normally otherwise do. All right, so we're going to move on to the thyroid gland here. The thyroid gland is shaped like a butterfly. It's located just inferior to the larynx and just anterior to the trachea, so just below the larynx and just in front of the trachea. And it has both a left and a right lateral lobe connected by an isthmus, and some individuals also have a pyramidal lobe projecting from that isthmus. It's not depicted here. Now, follicular cells inside the thyroid gland basically are stimulated by thyroid-stimulating hormone to make T3 and T4. These are the thyroid hormones. They're known as thyroxine and triiodothyronine. Again, they're the thyroid hormones. And we also have parafollicular cells in this region, which are right outside of the follicle, produce the hormone calcitonin, and that's going to be involved in the regulation of calcium homeostasis. So that looks kind of like this if you're looking at the thyroid gland itself. So these here are the parafollicular cells. They're going to be found on the outside of the follicle, these guys here. This whole thing here is a follicle. And then inside the thyroid follicle, we're going to have little colloid thyroglobin. And again, if we're looking in the individual, here's the pyramidal lobe, here's the right lateral lobe, there's the left lateral lobe, found just underneath the hyoid. Right here's the hyoid bone here, just underneath the larynx, which is underneath that. Let's see. And the way that T3 and T4 are synthesized is not something that I'm going to quiz you on, but I will walk you through it now. And I want you to basically, what I want the takeaway from this slide to be is that there are multiple different steps, and these compounds go through many different stages. First, they are going to be secreted from So first we have iodine trapping as the very first step. Iodine comes in, then iodine gets oxidized. At the same time that we're secreting this TGB, which is an intermediary molecule. So that synthesis and secretion of TGB happens at the same time as the oxidation of iodide. And then we have the iodinization of tyrosine, which is a specific region on this molecule that's going to end up with an extra iodine molecule on it. From there, T1 and T2 are going to couple together and then get they're going to get endocytosed back into this cell in a lysosome. And then that becomes T3 and T4, which are then secreted back out into the blood plasma. So there is an initial synthesis that happens inside these cells here, and then they get secreted into the lumen. In the colloid region, they undergo several major steps: oxidation of iodide, the iodinization of a tyrosine molecule on the TGB, and then T1 and T2 get coupled and then reabsorbed back in and then secreted into the bloodstream. So that's what the thyroid gland does, is it creates T3 and T4 from precursor molecules through multiple different steps. But what do thyroid hormones do? So thyroid hormones are involved in the basal metabolic rate and the maintenance of your normal body temperature. They're involved in the stimulation of protein synthesis. They are involved in the use of glucose and fatty acids for the production of energy via the creation of an ATP molecule. They upregulate beta receptors that attach to catecholamines, and they work with human growth hormone and insulin to accelerate the body growth. So they have multiple different functions in regulating different things of the body. Now, thyrotropin releasing hormone and thyroid stimulating hormone, they come from two different regions, right? TRH from the hypothalamus and TSH from the anterior pituitary, are going to help stimulate the synthesis and release of those thyroid hormones. It happens in a 5-step process that looks a little something like this. So if we have low levels of T3 or T4, that's going to stimulate thyroid releasing hormone. It can also be stimulated by a low metabolic rate. So if we want to increase our metabolism, we can also upregulate this pathway. So this releasing hormone, which is carried from the hypothalamus to the anterior pituitary, and it's carried by the hypophyseal portal vein system, stimulates the release of thyroid stimulating hormone by thyrotropes. Once we have thyroid stimulating hormone in the blood, it's eventually going to reach the thyroid follicular cells. So it's going to happen by way of the blood supply in the thyroid follicle, which is going to help go through all of those steps that I just talked about one at a time to be able to make T3 and T4 in the follicle, which then get released into the blood supply, which is then going to elevate the levels of T3 and T4 in the blood, which in this case, T3 and T4 are their own inhibitors. So they end up inhibiting the pathway. And some actions of the thyroid hormones, as I just mentioned, they're involved in the basal metabolic rate. They increase your body temperature, stimulate the synthesis of proteins, increase the use of glucose and fatty acids to create ATP, break down lipids, and then also regulate growth and development of your nervous tissue and your bones. So it's very important, and individuals who have issues with the thyroid or imbalances with the thyroid hormones, oftentimes it's as simple as taking, once they figure out what's wrong with the metabolic pathway, taking exogenous supplements, so thyroid medicine, to be able to get normal levels of whatever it is that they're lacking from these pathways. All right, so again, the thyroid gland is going to secrete these two major types of hormones. So T3 and T4 are thyroid hormones. They come from the follicular cells, right? And then also calcitonin, which comes from the parafollicular cells, which sit on the outside of the follicle. T3 and T4 are going to be controlled by thyrotropin releasing hormone, or TRH. TRH stimulates the release of TSH, and this is going to happen if we have low thyroid hormone levels, low metabolic rate. It can also happen with pregnancy, cold temperatures, high altitudes. And TRH and TSH are going to be inhibited by high thyroid levels, so high T3 or T4 levels is going to inhibit TRH and TSH, so kind of like an auto-inhibitory pathway. And then high iodine levels can also suppress the secretion of T3 and T4. And I already talked about the principal actions of the thyroid hormones a minute ago, so I'll skip through that. Now let's talk about calcitonin. Calcitonin comes from those parafollicular cells, and it is under the control of the concentration of calcium in the blood. If we have high levels of blood calcium, that's going to stimulate the secretion. If we have low levels of blood calcium, that's going to inhibit the secretion. And really, what's happening here is, in order to maintain appropriate concentrations of calcium in the blood, we are affecting the calcium that goes into the bone. So if we have too high of a level of calcium in the blood, we're going to store it in the bone, which would be like kind of putting money in the bank. And if we have too low levels of calcium in the blood, we're going to withdraw it from the bank and take it out of the bone, if that makes sense. Okay, so low blood levels of calcium are going to inhibit the reabsorption by osteoclasts and accelerating uptake of calcium. If we have high levels and we want to lower the levels, we'll inhibit bone resorption by osteoclasts and accelerating uptake of calcium and phosphates into the bone extracellular matrix. So basically, if we have too much blood calcium, we're going to deposit it into the bone, into the bone bank, basically. Okay, so we also have parathyroid glands. Now, parathyroid glands are located just behind the thyroid glands. We have one inferior and one superior parathyroid gland on each side. So here's our left superior and our left inferior parathyroid. Here's our right superior and our right inferior parathyroid. Parathyroid glands have two different types of cells, chief cells and oxiphyll cells. Now, chief cells, also known as principal cells, produce PTH, that's parathormone or parathyroid hormone. And oxiphyll cells have a not quite known function in normal pathways, but it is known that they secrete high amounts of PTH in cases of parathyroid cancer. So when they go awry, they secrete a lot of parathyroid. This is a light microscopic image of a parathyroid gland. Here's a vein or venule and an artery or arteriole, and then here's the blood capillary. These guys here are our chief cells, and these are our oxiphyll cells. All right, so calcitonin, which is produced by the thyroid glands, is going to work hand in hand with PTH, or parathyroid hormone, and calcitriol. So all of these work hand in hand to regulate the concentration of calcium in your blood. Okay, so I'm going to start with 1 and then go down to 2, just so you're aware of the lining here. So we're going to start here with the high levels of calcium in blood. So if our bloodstream has too much calcium in it, that's going to stimulate the release of calcitonin, or CT. If we have high levels of CT, that inhibits the osteoclasts, which is going to decrease the blood calcium level. It basically inhibits the ones that are going to be breaking down the bone to release calcium. Means we're going to be putting more calcium in than we're taking calcium out. That's going to lower the levels of calcium in our blood, which may end up getting too low, and low levels of calcium in the blood are going to stimulate the release of parathyroid hormone from the chief cells. When we have the release of parathyroid hormone, that's going to promote the release of calcium, again, from the bone, into the blood supply. And also, secondarily, it's going to slow the loss of calcium in the urine. And also, by way of the release of calcitriol, we're going to help stimulate the absorption of calcium from the food. So when we need calcium, we're not only going to get it from the bone, but we're also going to try to get it by turning off whatever calcium might be lost in the kidney. So resorbing that calcium, and then also resorbing as much as possible or increased absorption of calcium from the foods. All of this will result in an increase of your blood calcium level, which brings us back to 1. So again, the objective is to maintain homeostasis. Too high calcium, we need to deposit it in the bone bank. Too low calcium, we need to retrieve from the bone bank, right? Okay, so this is an image showing the chief cells in the parathyroid gland. And again, they're going to secrete parathyroid hormone. And it's going to be controlled by the levels of calcium in the blood. If we have low levels of calcium, we're going to stimulate secretion of parathyroid hormone. If we have high levels of blood calcium, it's going to inhibit the secretion of parathyroid hormone, right? And again, the action of the parathyroid hormone is to increase the calcium and magnesium in the blood and increase the bone resorption by osteoclasts. So we want to make sure that we're either, if we have low blood levels, we want to make sure we're taking money out of the bone bank. And if we have high blood levels, it's going to inhibit the secretion, which is going to increase the amount of money or calcium that is put back in the bone bank. Right? It's also going to increase the rate of calcium that is resorbed back from the urine and absorbed from the food. Okay, so let's talk about the adrenal glands for a second. Adrenal glands are also called suprarenal glands because they are found just above the kidneys. And here's our right adrenal gland and our left adrenal gland. And they're going to be serviced by the inferior phrenic arteries. Again, they lay right on top of the kidneys. So they're divided into 2 major regions. the outer cortex and the middle medulla. And you'll actually see this in several different sets of tissues. So this is not the only set of tissues to have a cortex and a medulla. The kidney also has a cortex and a medulla, same as the testes. So cortex is just going to be the outer layer, and the inner layer is the medulla. Now, the whole set of these glands is going to be covered by a connective tissue capsule. So that's depicted here in yellow. Now, if we're looking at the cortex, again, that's the outside. Histologically, it's divided into three major regions. That would be first the zona glomerulosa, the zona fasciculata, so glomerulosa up here, fasciculata, and then on the bottom, the zona reticularis. The zona reticularis secretes androgens. The fasciculata secretes glucocorticoids, mainly the glucocorticoid cortisol. And the zona glomerulosa secretes mineralocorticoids, mainly aldosterone. If we keep going down into the medulla, right here, now we have the adrenomedulla chromaffin cells. Remember, they're responsible for the secretion of epinephrine and norepinephrine when acted upon in the autonomic nervous system, right? We talked about that last lecture, I guess 2 lectures ago. So again, the zona glomerulosa secretes mineralocorticoids. These are hormones that are involved in the regulation of homeostasis in terms of the mineral levels. The zona fasciculata secretes glucocorticoids, and these are going to affect glucose homeostasis, so the amount of glucose in the blood. And the zona reticularis is going to secrete androgens, which are going to be masculinizing hormones. The most prominent mineralocorticoid secreted by the adrenal gland is aldosterone. It's involved in the regulation of sodium and in the homeostasis of potassium. And also, the adrenal glands are involved in the renin-angiotensin-aldosterone pathway, which controls secretion of aldosterone. And when we talk about kidneys and kidney function, we'll talk about this. It's very important in the water pathways, the water reuptake pathways. All right, so that looks kind of like this as an overall. So say that something happens to your body that is going to lead to a decrease in blood volume. Now, this could be dehydration. It could be a sodium deficiency, or it could be a hemorrhage, right? You've sliced your arm open or something and you've lost a lot of blood. And that's going to lead to a decrease in blood volume, which leads to a decrease in blood pressure. All right? So when we have a decrease in blood pressure, that's going to be detected by the juxtaglomerular cells of the kidneys, which are going to increase the levels of renin. At the same time, the liver is going to be reacting to that decrease in blood pressure by creating something called angiotensinogen. Anytime you see inogen, it's going to be an inactive version of something. So angiotensinogen gets activated to make angiotensin, if that makes sense. And renin and angiotensinogen together are going to increase the levels of angiotensin 1. Now, in the lungs, we have an enzyme called angiotensin-converting enzyme, which is going to change angiotensin 1 into angiotensin 2. And angiotensin 2 is going to act on several different parts of the body, including the arterioles and the adrenal cortex. So when it acts on the adrenal cortex, that's going to increase the secretion of aldosterone. And aldosterone is involved in water resorption. So increased aldosterone means that the kidneys are going to increase their sodium and water resorption. And by contrast, they're going to increase the secretion of the hydrogen ions and potassium ions into the urine, right? And all of that's going to lead to increased blood volume. Why? Because we are resorbing the water from the kidney or from the urine, so it's going to go back into the blood. Additionally, if we look at 15 here, not only are we affecting the adrenal cortex, but in the arterioles, we're going to have vasoconstriction or tightening, which is also going to increase blood pressure. And that's going to continue until blood pressure returns back to normal. So this whole pathway is involved in making sure that we have appropriate levels of blood pressure in extreme situations like hemorrhage and dehydration. Now, glucocorticoids, such as cortisol and hydrocortisone, cortisone and corticosterone, all of these are secreted and regulated by negative feedback loops and are responsible for helping maintain and control the breakdown of protein, the formation of glucose. Lipolysis is the breakdown of lipids, also resistance to stress, and the inflammation and immune responses. Again, so here is a negative feedback loop. In this case, we have a stimulus that is disrupting a controlled condition. The controlled condition in this case is glucocorticoid levels in the blood. And they're going to be detected, right? The change has to be detected by some sort of receptor. In this case, the receptor are the neurosecretory cells in the hypothalamus, which are then going to be sending their information on in terms of input to a control center. The control center in this case is not the brain. It's not always the brain. Here's the corticotrophs in the anterior pituitary, which are then going to increase ACTH. The increase of ACTH is going to affect the adrenal cortex in form of the secretion of glucocorticoids. Remember, we had a low glucocorticoid level in the first place, so that's going to increase the glucocorticoid level in the blood, hopefully back to the point where the glucocorticoid level is going to return back to normal, right? And this is an example of a very classic negative feedback loop. Now, the adrenal glands also secrete androgens, and the major androgen that's secreted, again, by the adrenal cortex is going to be DHEA, or dihydroepiandrosterone. This is secreted in males, and after puberty, the hormone testosterone is secreted in much larger quantities, so DHEA has much less of an effect. So the main role that DHEA plays in males is going to be pre-puberty. In females, we do have DHEA and other types of androgens, so it's a very common misconception that women only get female hormones and men only get male hormones. And in reality, it's a combination of the both, and female get more female hormones than males, and males get more male hormones than females, but we both get both, and they regulate multiple different biological processes, a little bit beyond what you think of when you think of just sexual differentiation and development. In females, DHEA plays a role in promoting libido and also is able to be converted into estrogens. So androgens are able to be converted into estrogens and vice versa. And in fact, in menopausal women, all the female estrogens in their body are going to come from adrenal androgens. They're no longer producing any estrogens. They're just converting adrenal androgens into estrogens after menopause. Now, we talked about the adrenal medulla when we talked about the autonomic nervous system, but just to kind of brush up your memory here, there are chromaffin cells inside the adrenal medulla that secrete adrenaline, or epinephrine, and norepinephrine, which is known as noradrenaline, and these are involved in the sympathetic pathways, right, in the fight or flight response pathways. Okay, so this summarizes the different adrenal gland hormones. So in the cortex, we have mineralocorticoids, namely aldosterone, and they're secreted from the zona glomerulosa cells involved in the regulation of potassium levels and angiotensin II levels. They're controlled by the level of blood calcium and angiotensin II levels. These are both going to increase the secretion of these mineralocorticoids, and their actions are that they're going to increase the levels in the blood of sodium and the water and decrease the blood level of potassium. Glucocorticoids, namely cortisol, they come from the zona fasciculata. That's going to be under the control of ACTH. ACTH will increase the release. Corticotrophin-releasing hormone is going to produce the secretion of ACTH in response to things like stress and low blood levels of glucocorticoids. The actions of glucocorticoids are to break down protein, with the exception of in the liver, stimulate gluconeogenesis, or the creation of new glucose, to stimulate lipolysis, or the breakdown of lipids. All of this would be to be able to provide energy to the body. It's also going to be involved in inflammation, immune responses, and stress responses. Androgens that are found in the adrenal cortex, they're found from the zona reticularis cells. Mainly, we're talking here about DHEA, which is going to be stimulated by ACTH. And its objective in men is going to be the early growth of axillary and pubic hair in both sexes. Again, after males hit puberty, testosterone takes over, so DHEA doesn't have as much of an effect anymore. And females responsible for contribution to libido and after menopause as the only source of estrogens. So the adrenal androgens are going to be the only source of estrogens post-menopause. And then last but not least, the adrenomedullary hormones include epinephrine and norepinephrine. They come from those chromaffin cells, and they are under response from the sympathetic preganglionic neurons, which release acetylcholine, stimulation of the secretion. And all of that, if you need a refresher, you should go back to the autonomic nervous system lecture. The effect of this would be to enhance the effects of the sympathetic division of the autonomic nervous system. So basically, it's going to allow you to have an increased fight or flight response. All right, so let's talk about the pancreas for a little while. The pancreas is both an endocrine and an exocrine gland. So it both secretes hormones into the bloodstream and secretes non-hormonally directly into different ducts, into the intestines. It's located in the curve of the duodenum, and I know we haven't quite gotten into digestion yet, so that might not be familiar to you, but the duodenum is part of the intestines, actually of the small intestines, and it's going to help by release of intestinal enzymes. So here's the head of the pancreas here. Come on, head of the pancreas here, and the tail of the pancreas, which is going to go right into the spleen. They're both going to be fed by the splenic artery on this side and by the hepatic artery on the other side. Here we have the superior mesenteric artery, and then this is going to be the celiac trunk of the abdominal aorta. So this is going to be the aortic arch, or head into the aorta, sorry. Head up, not into the aortic arch, I apologize. And the abdominal aorta, which runs down from the aortic arch. All right, so pancreatic islets. So the pancreas has exocrine cells that are arranged in these little clusters. The clusters are called acini, and they just produce a bunch of digestive enzymes that are then going to get delivered into the intestines, into the gastrointestinal tract through ducts. And we have different regions here that are called pancreatic islets, or islets of Langerhans, because he's the one that discovered them. And they're scattered in among the acini, and they're actually going to be endocrine tissue. So this looks kind of like that. So if we take a light microscope and we are going to actually take a look, here we have a pancreatic islet. So here we have it at 40x, and then if we're going to zoom in on it, here we have the pancreatic islet. Inside there we have beta cells and alpha cells. And if we look at this depiction over here, here's the pancreatic islet and the surrounding acini, and we have the blood capillaries depicted here in red, also alpha cells, which are responsible for secreting glucagon, beta cells, which secrete insulin, delta cells, which secrete somatostatin, and F cells, which secrete pancreatic polypeptides. So we're secreting a lot of different what can be considered digestive enzymes, or at least digestive regulatory molecules. Again, the islet contains 4 different types of cells that secrete different types of hormones. Alpha cells secrete glucagon, beta cells insulin, delta cells secrete somatostatin, and F cells secrete pancreatic polypeptide. So if we're going to look at the hormone themselves and the source, here's glucagon. Again, it's from the alpha cells. The control of that is under the control of the glucose levels. So if we have a low level in our blood of glucose, it can also be under the control of exercise or meals that are mainly protein, which aren't going to provide a lot of glucose into your bloodstream. And then it's inhibited by somatostatin and insulin. The actions of glucagon is to increase the blood glucose level, and it does that by increasing the breakdown of glycogen into the glucose, glycogen into glucose in the liver, and that's called glycogenolysis, or lysis of glycogen. And we can also turn things into glucose with gluconeogenesis, or creation of new glucose, and that'll help release glucose into the blood supply. The next hormone to look at is insulin. It's secreted from the beta cells of the pancreas and the islets. It's under, again, the control of the blood level of glucose, but it's basically under the control of an increased blood level. So if we have increased blood level of glucose, or increased acetylcholine, or increased glucagon, all that's going to stimulate secretion, and it's going to be inhibited by somatostatin. And it's going to work kind of the opposite way from glucagon, in that it's going to lower blood glucose levels by accelerating the glucose transport into the cells, also by converting glucose into glycogen. That's glycogenesis, which is the exact opposite of glycogenolysis, right? Glycogenolysis is breaking down glycogen. Glycogenesis is the creation of new glycogen, and also decreasing glycogenolysis. So that's the flip side of the same coin. If you're undergoing glycogenesis, you're not going to also be undergoing glycogenolysis, because they're basically equal and opposites. And it's also going to increase lipogenesis, or the creation of lipids, and stimulate protein synthesis. So it's going to work almost exactly opposite of glucagon. We also have somatostatin, which is found from the delta cells of the pancreatic islets. And it's under the control of pancreatic polypeptide, which is what's going to inhibit its secretion. And its role is to inhibit the secretion of both insulin and glucagon in order to slow absorption of nutrients from the gastrointestinal tract. We also have pancreatic polypeptide. These are made by the F cells, and they're under the control of, so the secretion would happen if we had meals containing high protein, or if we had a lot of fasting, which meant that we weren't going to be bringing in much blood glucose, or exercise. So high exercise might mean we're running through our metabolites very quickly. Also acute hypoglycemia, which would be low glycemic index, and that's going to stimulate the secretion of this pancreatic polypeptide. So somatostatin and elevated blood glucose levels are going to inhibit it. So if you have high glucose levels, you won't need it. And what does it do? It inhibits somatostatin secretion, which is interesting because it's inhibited by somatostatin. So these two kind of like work hand in hand. It inhibits somatostatin secretion and gallbladder contraction and the secretion of the pancreatic digestive enzymes. Okay, so all of these are controlled by a negative feedback. So again, we're going to start at 1 with the low blood glucose. That's again hypoglycemia. If we have low blood glucose, that's going to stimulate the alpha cells of these islets to secrete glucagon. When we have glucagon, that's going to act on the liver cells in order to convert glycogen into glucose, also to form glucose from lactic acid and other certain amino acids, so gluconeogenesis. And the glucose released by the liver cells is going to increase the blood glucose level, hopefully to normal, sometimes too far. So if we end up with hyperglycemia, now we're too high, and that's going to inhibit the release of glucagon and also stimulate the beta cells to secrete insulin. So again, anytime we go too far to the left, we want to bring it back to the center, too far to the right, same thing, bring it back to the center. So if we have high blood glucose levels, not only is it inhibiting the release of glucagon, so it's inhibiting this pathway, but it's also stimulating this other pathway over here, stimulating the release of insulin from the beta cells, which does the exact equal and opposite, accelerated facilitation of diffusion of glucose into cells, so bringing the glucose out of the bloodstream into the cells, converting glucose into glycogen, and increasing the uptake of amino acids to increase protein synthesis and also speed the synthesis of fatty acids. Now, if we do this, all of that will help bring the blood glucose level back down. And if the blood glucose level continues to fall, we can end up in hypoglycemic, which is going to inhibit the release of insulin and lather, rinse, repeat, increase the secretion of glucagon, right? So these two are controlled by kind of interconnected negative feedback loops. Okay, so let's talk about some organs that also create hormones, such as the gonads, or the ovaries in females and the testes in males. So their objective of these organs is to produce gametes, producing oocytes in females or eggs, and sperm in the testes. And ovaries produce 2 different estrogens, including estradiol and estrone. It also produces progesterone, relaxin, and inhibin. And the testes produce testosterone. So these are all hormones that are created by these organs. All right, so ovarian hormones are going to include estrogens and progesterone, and they're going to work in coordination with other gonadotropic hormones of the anterior pituitary to help regulate the female reproductive cycle and maintain pregnancy, prepare the mammary glands for lactation, and then also promote the development and maintenance of female secondary sex characteristics. So basically, the induction of the female sex. Relaxin is involved in pregnancy whereby it helps increase the flexibility of the pubic symphysis. It also really relaxes a lot of different joints, so ankles can also become wobbly, but this is going to help allow the pubic bones to pull apart during delivery. It's also going to help with the dilation of the uterine cervix during labor and delivery. And inhibin is going to be responsible for inhibiting the secretion of FSH, or follicle-stimulating hormone, from the anterior pituitary. And this is obviously in a timely fashion because sometimes we do want FSH. All right, so in the male, we have testosterone that's going to be secreted from the testes, and it's going to help stimulate testicular descent before birth, regulate the production of sperm, and promoting the development and maintenance of the male secondary sex differentiation and development. And again, inhibin is going to help inhibit the secretion of FSH from the anterior pituitary. So now we're going to move on to the pineal gland and the thymus. The pineal gland is actually attached to the roof of the brain in the third ventricle, and it secretes melatonin, and melatonin is involved in regulation of the biological clock. The thymus is going to be located behind the sternum, between the lungs, and it's going to produce thymic humoral factor, thymosin, thymic factor, and thymopoietin, all of which are going to be involved in the maturation of the immune system cells, specifically of the T cells. So they're going to help create T cells. Okay, so the pineal gland is going to be found back here. Right, so if here's the hypothalamus and the pituitary gland, the pineal gland is located in the back. In the ventricle. We also have other tissues and organs that are not part of the endocrine system that are able to produce hormones. Here's some examples of those. So the gastrointestinal tract, for example, can secrete gastrin, also can secrete glucose-dependent insulinotropic peptide, and CCK or cholecystokinin. All of these are involved in digestion, so they're involved in the secretion of gastric juices, for example, or the release of insulin by the beta cells. And secretin is going to stimulate the secretion of pancreatic juices and bile. So all of these are going to be hormones that are involved in regulation of digestion. And here's a temporary organ called the placenta. The placenta also creates its own hormones. In fact, for a little while, it is the actual hormonal control and regulation of lots of what's going on in the female body, as well as in the developing embryo, well, fetus. And that's going to involve the secretion of human chorionic gonadotrophin, or HCG. Anyone who's ever taken a pregnancy test knows that this is the molecule that you're actually testing for. Low levels mean you only get one line on the stick. High levels mean you get two lines on the stick. And it's going to be involved in stimulating the corpus luteum of the ovary, which is the region after. So the follicle bursts open, the egg pops out, and then goes into the oviduct, and with any luck ends up fertilized and implanting in the uterus. If it implants in the uterus, the corpus luteum is responsible for secreting hormones for a little while, and that's going to happen but via the human chorionic gonadotrophin. So the corpus luteum secretes this HCG, which again is what you detect in your pregnancy tests, and it's going to help maintain pregnancy, particularly in the early stages until the placenta is able to take over. Estrogens and progesterone are involved in the maintenance of pregnancy and also helpful in creating the milk and secretion of the milk from the mammary glands. Human chorionic somatotropin is involved in the development of the mammary glands for lactation as well. So it's going to have an increase of the breast, and the swelling of the breast is going to allow for more lactation to occur during breastfeeding. And in the kidneys are going to secrete renin, erythropoietin, and calcitriol. Renin is involved in regulation of the blood pressure. So it's going to be part of vasoconstriction and secretion of aldosterone, or we talked about how that was also involved in water reuptake, etc. And we'll get an entire lecture on that when we get to the kidneys. So I'm just kind of glossing over it here. Erythropoietin, you might recognize erythro from erythrocyte. Poietin means creation of, so erythropoietin is going to increase the rate of red blood cell creation. Calcitriol is going to help absorb dietary calcium and phosphorus. And in the heart, we're going to be making ANP, or atrial natriuretic peptide, and that's going to help in regulation of blood pressure, namely to decrease the blood pressure. And in adipose tissue, we make leptin. Leptin is, again, created by fat cells and is involved in hunger and satiety. It helps give you a feeling of being full or suppressing your appetite. And it's also involved in stimulation of FSH, or follicle stimulating hormone, and luteinizing hormone activity. Okay, so let's talk about echinosoids. These are hormones that are locally acting, so instead of being globally distributed, they're going to act in a local area, and they're derived from arachidonic acid. And these are going to basically fall into the category, they all have 20 carbon fatty acids, etc. They fall into their own category, and they often are going to be involved in the stimulation of cell growth and division. We also have a lot of different growth hormones. So growth factors, these are known to be involved in tissue development, growth, and repair. So here's some examples of different growth factors. It's going to include epidermal growth factor, platelet-derived growth factor, fibroblast growth factor, and as you can imagine, this is going to help create more epidermal tissue. This is going to help create more platelet tissue, more fibroblast tissue, more nervous tissue. Tumor angiogenesis factors, TAFs, these are going to help create more blood vessels. So angiogenesis is the formation of new blood vessels, and one of the first things a tumor needs is a really good blood supply to latch onto. And while it's produced by normal cells, it's upregulated in tumor cells, and that's how they discovered it. So that's why it's called tumor angiogenesis factor. And basically, it's going to be stimulation or induced stimulation of new capillaries and help with organ regeneration and wound healing. And transforming growth factors are basically going to be very similar to like epidermal growth factor, etc. So it's going to help increase epidermal growth for alpha, and TGF beta is going to inhibit proliferation of different cell types as well. So that's, TGF beta is going to help basically have a check on proliferation of many cells. Okay, so let's discuss the stress response. So everybody is constantly under a little bit of stress. You're under stress right now because we have an exam coming up, right? Or you're a little bit stressed because you have to make it to the grocery store. So eustress is the normal everyday stress. That's the get up out of bed and go get stuff done stress, right? Without eustress, you would never do your dishes because you would have no worries about the dishes. So eustress is helpful everyday stress that helps you meet the challenges of your life. Now, we also have harmful types of stress. Any type of stress that may be damaging, including distress. So this would be if you have pushed your body to a limit, you've ended up injured, or something has happened emotionally that has caused damage to your body. So distress is a negative type of harmful stress. And we also have the fight or flight response, which is, again, the sympathetic nervous system. This is the very first stage of the stress response. And the very first thing that happens is it's going to stimulate the body's resources to prepare for immediate activity. And you may recognize when we talked about the autonomic nervous system, some of these sympathetic responses are going to include increasing your blood pressure, increasing your heart rate, increasing your blood glucose, all things that are increasing your ATP levels, all things that are going to prepare your body for immediate fight or flight. The second stage of the stress response is the resistance reaction, and this is a little bit longer. It's going to last quite longer than the fight or flight response. Okay, so this is an overview of the stress response. And I know there's a lot going on here, so we're going to walk through it very, very slowly. We have stressors that can stimulate. So any stressor can stimulate nervous impulses. Again, this is going to be the sympathetic pathway. It's going to go to the sympathetic spinal cord. Sympathetic centers of the spinal cord go out to the sympathetic nerves and affect visceral effectors, right? One of those things is going to be the adrenal medulla, whereby we secrete epinephrine and norepinephrine to start that fight or flight response. Again, that comes from the chromaffin cells. Some of the visceral effectors also are going to induce that stress response. It's also going to stimulate a decrease in CRH, GHRH, and TRH. Right? All of those are going to have their own, so CRH acts on ACTH, so decrease in CRH, right? That's a releasing hormone. Now we have less ACTH. If we have a decrease in GHRH, that gives us a decrease in HGH, right? So growth hormone releasing hormone. If we have less of that, then we have less human growth hormone released. Thyroid releasing hormone, we have less of that. We have less thyroid stimulating hormone coming out, right? So if we have a decrease in all of these from the anterior pituitary, that's going to act on multiple things like the adrenal cortex. And the stress response there would induce lipolysis, or the breakdown of lipids. You're also going to see that in response to the decrease in HGH and IGFs. You're also going to see gluconeogenesis, the creation of new glucose molecules, because again, we're going to need that glucose in our blood to be able to meet whatever the challenges are. We'll increase protein catabolism, sensitization of your blood vessels, and reduction of inflammation. In terms of human growth factor, less human growth factor and less IGFs are going to result in lipolysis. Again, the release of breakdown of lipids, so we have immediate available energy sources, and then glycogenolysis, which is breaking down glycogen to create glucose. All this will increase our blood glucose levels. And then last but not least, if we have a decrease in thyroid stimulating hormone, that's going to decrease these thyroid hormones, which is going to increase the use of glucose in order to produce more ATP. Again, all of these responses are so that you're able to meet the needs of fight or flight. Increased ATP, increased blood glucose levels, increased fatty acids in your blood to be able to use for energy, right? All of these are going to help give you an immediate response to be able to meet the needs of fight or flight. Okay, so for the purposes of our exam, development is never on here, so I'm going to gloss through this. This is really only here in case you have any interest in taking a developmental biology course, which I highly recommend, but I will not test anyone on the development of the endocrine system. So let's talk a little bit about aging. As we get older, the hormone levels are going to change. Some of them are going to increase, but some of them are going to decrease. And we also are going to have levels of some things that are going to remain the same, epinephrine, norepinephrine, etc. If you look on a histological basis, so if you're looking at slides of these glands, most endocrine glands are going to get smaller and become more fibrous as you get older. So they're going to have less and less functional surface area. And this slide just talks about homeostasis and how the endocrine system is going to contribute to all of these different functions within the body. So I really appreciate you taking your time. Aloha, and I will see you all next time. Thank you very much for listening, and have a great day.